Hi everyone, welcome to this brief introduction to target identification, validation and hit finding at WuXi AppTec. My name is Dave Madge, I'm based in Cambridge in the UK and support our client base in Europe. It's great to have this opportunity to talk to you. To give you a brief introduction to WuXi AppTec, we're a global organization providing support and collaboration to our pharma, biotech and academic partners from 30 sites spread across 9 countries. Our headquarters are in Shanghai, but as you can see, we also have substantial operations in America and Europe. Today, we support over 4,000 partners. Our philosophy is to provide an end-to-end platform that enables all kinds of partners to develop their therapeutic discovery and development projects faster by removing barriers to entry for all technology platforms and providing world-class resources. Within the WuXi AppTec organization, we support small molecule, peptide, and oligonucleotide discovery and manufacturing, as well as cell and gene therapy, medical device testing, and clinical research. Our target to IND platform supports all stages of oncology and immunology drug discovery, from target ID and validation through hit finding, hit expansion, and lead optimization. Along the way, there's a comprehensive suite of tools and technologies that can be accessed according to project needs to accelerate this discovery pipeline. I'll talk about some of these platforms in more detail in the following slides, but the main point I want to make on this slide is the depth of the platform and the wide range of services and technologies available throughout the discovery process. So to turn to oncology and immunology in more detail. Of course, the first thing we know about this area of drug discovery is the importance of finding new therapies and the substantial impact on human health that success in this area has generated, especially over the last 10 years or so. The diversity of therapeutic modalities and the rapidly increasing number of targets under investigation creates a deep pipeline of new research and clinical entities. We've been privileged to be able to support many discovery and development projects in this area, and I'd like to discuss some of the tools we've developed along the way. To provide a roadmap for this discussion, these are the core disciplines that I'd like to comment on. We'll start with target ID and validation, in particular referencing genetic tools, then go on to discuss first in vitro and then in vivo assays, and finally talk a little about biomarkers. So on this slide, I'm showing the core building blocks of the target ID and validation platform, encompassing an advanced gene manipulation platform, a large, diverse cell bank of over 1,000 cancer cell lines, mouse genetics, and in vivo gene editing, and of course, a wide range of disease models. To focus now on the gene manipulation platform, this encompasses bespoke cell line generation, including knock-in and knockout cell lines, sgRNA library screens for synthetic lethality, and drug modifier screens. We work with primary cells, including immune cells and stem cells. And we can use base editing and prime editing and build phenotypic assays, including high content analysis. And of course, we can support targeted protein degradation, including high-bit, DTAG, and SMASH platforms. With respect to CRISPR tools, we work with knockout, repression, and activation libraries, looking for essential genes in dropout or synthetic lethal modes. We can look for genes that have become essential under particular selection pressures or confer survival advantages. Across these different modes, we're running 15 to 20 screens per year. So there's a wealth of experience with these assays, reducing assay development times and maximizing confidence in the data generated. We show on this slide a little of the workflow involved in some of these screens, especially synthetic lethal and drug modifier screens. Here we're using selector shRNA and sgRNA libraries, with these libraries covering signaling pathways, disease-associated genes, cell surface, extracellular, and DNA binding targets, as well as enabling whole genome screens. We've run these, as shown here, in synthetic lethality and drug resistance or sensitivity screens, followed by next-generation sequencing and data analysis. On this slide, we show a little data where we've investigated both single and double knockout screens in cancer cells using sgRNA design and expression, polyclonal cell characterization, followed by single clone selection, functional analysis, and characterization of the resulting engineered cell lines. We also have a robust workflow for identification and validation of drug resistance mechanisms, wherein we use RNA-seq to identify candidate-resistant genes by profiling selected resistant cells, followed by CRISPR to knock in mutations into parental cells, and then looking for a potency shift for reference drugs in those mutant cells. To further improve the precision of the gene editing platform, we've implemented CRISPR prime editing, exemplified here for a his tag insertion into a HEC3 locus. To broaden the utility of our gene manipulation platform, we've successfully extended the implementation to various human and mouse primary immune cells, validated by sequencing and cell sorting. This enables multiple functional assays in modified primary cells, including RT-PCR and ELISA for cytokine measurements. On this slide, we show some of the scope of this application in mouse and human T cells, human monocytes, dendritic cells, and macrophages. With targets identified and validated, we may want to start looking for small molecule starting points for a drug discovery program. To support this, we built a comprehensive lead generation platform encompassing biochemical and phenotypic high throughput screens, affinity selection of small molecules by mass spectrometry, screening of DNA encoded libraries, fragment screening, and virtual screening. Each year, we run hundreds of hit finding campaigns using one or more of these platforms, and can provide fragment libraries, HTS decks, and DNA encoded libraries to support these activities. To further support optimization of hits generated using the platforms just described, we have available a wide range of medium throughput assay formats that will typically be used alongside a medicinal chemistry program. These screening formats support all target classes and are backed by a comprehensive suite of liquid handling, compound dispensing, and data management and analysis tools. I'd like now to turn to in vitro efficacy studies that can be carried out on the various cell types that contribute to the tumor microenvironment. In particular, I'll briefly present some assays we support in T cells, macrophages, and NK cells. And I'll go on to show some data for one of the frequently studied targets in this area. This slide shows some data analyzing the mixed lymphocyte reaction in T cells and observing the dose-response effect of an anti-PD-1 antibody on cytokine production, including in the lower right panel, the effect of a combination of the PD-1 antibody and a small molecule ligand for another target in the pathway. We can also look at the impact of our compounds on the differentiation of helper T cells. For example, examining the relative distribution of Th1, Th2, Th17, and Treg cells as shown on this slide, with their corresponding cytokines. In macrophages, we might choose to examine the activation of M1 macrophages towards a more polarized phenotype, in particular M2 macrophages. We can visualize this split of a macrophage phenotype with FACS analysis, for example, looking at cluster of differentiation markers such as CD86 and CD163, as shown in the top right panel. And of course, we can also differentiate based on cytokine profiles, with IL-6 and TNF-alpha usually high in M1 macrophages, and IL-10 high in M2 macrophages, as shown in the lower right panel. Macrophage phagocytosis assays are commonly performed to examine the ability of an antibody for one of the so-called don't eat me signals, such as CD24 or CD47, to overcome these signals and trigger phagocytosis as shown on this slide. In this final slide on different immune cells, I'll briefly mention some assays from natural killer cells, as well as activation and cytotoxicity assays using FACS to distinguish, for example, CD56 and CD69 expressing NK cells. We can also look at effector to target, or ET ratios. On this slide, we're looking at cytotoxic effects on the Raji lymphoblastoid cell line in the top panel, and the effect of rituximab on cell lysis as a function of concentration and ET ratio in the lower panel. One of the most studied protein complexes recently is the NLRP3 inflammasome complex. NLRP3 is a critical component of the innate immune system, mediating caspase-1 activation and the secretion of pro-inflammatory cytokines. We have broad experience with this complex and have built a number of biophysical and biochemical assays to support drug discovery in this area. We've been able to produce NLRP3 protein complexes and their variants and validate their activity, and have gone on to develop, for example, nano-DSF, MST, and ADP glow assays to examine the effect of small molecules on complex stability, ligand binding, and ATPase activity, respectively. On this slide, we show the effect of the NLRP3 inflammasome inhibitor MCC950 on THP1 cells that have been stimulated with LPS and the inflammasome inducer, the gerasine. Moving closer to translational studies, our humanized models represent all major tumor types with extensive supportive sequencing and lymphocyte data, and have been used to investigate tumor growth inhibition, including tumor rechallenge and measurement of tumor infiltrating lymphocytes. We have collected extensive standard of care data with checkpoint inhibitors alone, alongside each other, and alongside other drug classes and treatment modalities. To help validate mechanism of action and to support biomarker discovery, we've developed profiling platforms for cell sorting, immunohistochemistry, macrophage migration inhibitory factors, and RNA sequencing. We also use nanostring technology to help identify gene disease associations in both oncology and immunology studies. We've been using human PBMC for adoptive transfer studies in mice. Shown here, for example, the functional T cells in these models release human cytokines. To date, we have developed 56 models using human PBMC adoptive transfer, including both CDX and PDX models, covering all the major tumor types and targets. To the right of this slide, you can see that we've also been using knock-in mice to express human target genes for checkpoint targeting antibodies, and demonstrating, for example, that CTLA4 and PD1 antibodies show substantial reduction in tumor volume in such models. Again, we have covered the major IO target genes and have built double and triple knock-in models, as well as paired syngeneic models in C57 and BOB-C backgrounds. To further extend the range of modalities we've characterized, I show here some data generated using bispecific T-cell engagers in the human PBMC adoptive transfer model. In this case, we're using an immunodeficient mouse model and looking at tumor growth using a bioluminescent readout under the IVIS system. You can see that the BITE compound, Blincyto, provides a robust inhibition of tumor growth, whilst two further test substances are much less profound in their effects. Finally, to support clinical biomarker development and application, we have a CAP-certified and GCP-compliant platform for tumor tissue processing, clinical pathology, and FACS analysis. We're using the HALO digital platform, and to date, we've validated more than 100 biomarkers. I'll finish here, but I'd like to thank you for joining me to view this presentation, and thanks also to the organizers for the invitation to speak. Please do get in touch if you'd like to learn more about any of the platforms we've discussed today, and thank you for your attention.